Welcome to this immersive educational journey into the expansive world of animal biotechnology. For this video, we'll delve deep into the definition of animal biotechnology and explore captivating examples that showcase innovation and transformative impact within this field. So, what is animal biotechnology? It is the art and science of producing genetically engineered animals. It is the new technology that opened many doors in animal farms, medical, and research field, as its course involves the application scientific techniques to manipulate the genetic material of animals. The field has redefined our relationship between with the animal kingdom, offering new possibilities in genetics, health, and reproduction. The term in vitro means glass or artificial conditions, and IVF refers to the fact of the fertilization of egg by sperm and occurred not in uterus but outside the uterus critically contained optimum condition. The IVF technology is very useful. It involves the procedure taking out the eggs from ovaries of female donor, in vitro maturation of egg cultures kept in an incubator, fertilization of the eggs in test tubes by semen obtained from superior male, and implantation of 7 days of old embryos in reproductive tract or other recipient female which acts as foster mother or surrogate mothers. These are used only to serve as animal incubator and to deliver offspring after normal gestation period. The surrogate mothers do not contribute anything in terms of genetic makeup since the same comes from the egg of donor mother and semen from artificial insemination. Our exploration begins in vitro fertilization, a revolutionary technique that allows fertilization to occur outside the animal's body. Witness how IVF has not only transformed human reproduction laws but has also been a game changer in selective breeding and genetic enhancement for various ant species. So next is artificial insemination. It is the transfer of a collected semen to a recipient female. Artificial insemination is the most valuable management practice to the cattle producer. In artificial insemination, a doctor inserts sperms directly into a woman's cervix, fallopian tubes, or uterus. The most common method is called intrauterine insemination or IUI, when a doctor places the sperm in the uterus. The procedure makes efficient of the generous supply of sperm available from an individual make in a manner that greatly increases genetic progress as well as improving reproductive efficiency in many situations. What is sex reversal in animals? Sex reversal is a redirection of sexual phenotype during embryonic development. Most aspects of sexual phenotype, including sex ducts and genitalia, depend on the presence of steroid hormones produced by testes or ovary. The testis or ovary fate of the gonad is decided through a process called sex determination. During sex determination, the testis and ovary pathways are eventually antagonistic and compete for control of gonad fate. Sex reversal occurs when sexual phenotype of the organism to the opposite sex. Moving on to artificial insemination, with sex reversal, a technique that extends beyond conception, explore how scientists manipulate sex of animals, contributing to more effective breeding programs and diverse applications in agriculture. Next is animal cloning. Cloning is the copying of animal gene into many copies. Start with embryo twinning or splitting embryos in half. Here's the advantage of cloning: preservation of endangered animals, studying the effect of drugs on duplicate improve the agricultural production. To make a clone, scientists transferred DNA from an animal's somatic cell into an egg cell that has had its nucleus and DNA removed. The egg develops into an embryo that contains the same genes as the cell donor. Then, the embryo is implanted into an adult female's uterus to grow. Now, let's delve into the intricate world of animal cloning from the iconic Dolly, the sheep of modern to modern advancements. Witness the fascinating processes involved in creating genetically identical copies and the implications for conservation, agriculture, and medical research. What is transgenic animals? It is a transfer of gene from one organism to another. Animals that have foreign gene inserted in their DNA, often for research or biopharmaceutical production. A transgenic animal is one whose genome has been changed to carry genes from another species to use technique for animal. Genome editing for specific traits. Animal features can be changed by purposely altering the gene or genes. A mouse was the first successful transgenic animal. Transgenic animals, marvels of genetic engineering, offer a glimpse into the future. Discover how introducing genes from one species to another can lead to animals with enhanced traits, disease resistance, and significant contributions to medical and agricultural fields. What is genetic engineering? It is a modification of an organism's characteristics by adjusting its genetic material. This can be done by old-fashioned breeding, transgenic, or by cloning. Altering an animal's genetic makeup to introduce specific traits or improve resistance to disease. Genetically engineered animals are also created to help medical researchers in their quest to find cures for genetic diseases like breast cancer. Finally, endangered animal species can be cloned. Trust thus helping wildlife management in its goals of preserving wild populations of the Earth's biological diversity and by ensuring endangered animals' genetic information will not be lost when the last of the species dies. Genetic engineering, a powerful tool allowing precise modification of an animal's DNA, explores real-world examples demonstrating how this technology addresses challenges, from disease prevention to developing animals with specific desirable traits. And what is embryo transfer? Embryo transfer, removing fertilized ova or embryos from donor and implanting in a recipient. Surgical and non-surgical methods are used to remove and implant. A quality donor female can produce more offspring. Embryo transfer is a simple technique and anesthesia or sedation is rarely required. A long thin catheter containing the agreed upon number of embryos along with a small amount of fluid is passed through the cervix into the uterus, where the embryo or embryos are released. Our journey concludes with the intricacies of embryo transfer, a delicate process crucial in animal breeding. Witness the strategic movement of embryos from donor to recipient, optimizing the genetic potential of valuable lines. Thank you for joining us on this in-depth odyssey. The journey into animal biotechnology is vast, and as we continue to unlock its secrets, may you remain at the forefront of discovery and innovation. Until next time, stay curious and embrace the wonders that science unfolds.